A 2-year-old child is brought to the emergency after an overnight fast with seizures and blood glucose is 28 milligrams per deciliter. But here's the twist, there are no ketones in urine. Why? The problem isn't glucose. The problem is beta-oxidation. Beta-oxidation is the mitochondrial pathway that breaks down fatty acids into acetyl-CoA along with NADH and FADH₂ to generate ATP. Before beta-oxidation begins, fatty acids must first be activated in the cytoplasm. In this step, a fatty acid reacts with coenzyme using energy from ATP. The reaction is catalyzed by the enzyme fatty acyl-CoA synthetase, also called thiokinase. The final product formed is fatty acetyl-CoA, which is now ready to enter the mitochondria for beta-oxidation. Long-chain fatty acetyl-CoA cannot cross the inner mitochondrial membrane directly, so the cell uses the carnitine shuttle. First, carnitine acyltransferase 1 (CPT-I) on the outer mitochondrial membrane transfers the acyl group from CoA to carnitine, forming acylcarnitine. This acylcarnitine is then transported across the inner mitochondrial membrane. Inside the matrix, carnitine acyltransferase 2 (CPT-II) converts it back to acetyl-CoA, releasing carnitine. Now the fatty acetyl-CoA is ready to undergo beta-oxidation. Inside the mitochondrial matrix, the beta-oxidation cycle begins. Each cycle removes 2 carbon atoms from the fatty acid chain. Step 1. Oxidation. 2 hydrogen atoms are removed from the alpha and beta carbons of fatty acetyl-CoA, forming a double bond between these carbons. These hydrogens are accepted by FAD, which is reduced to FADH₂. The product formed is enol-CoA. Step 2. Hydration. Next, the enzyme enol-CoA hydratase adds water across the double bond, producing beta-hydroxyacyl-CoA. Step 3. Oxidation. In the next step, hydrogen is again removed from the beta-hydroxy group, converting it into a keto group. These hydrogens are accepted by NAD, which is reduced to NADH, forming beta-ketoacyl-CoA. Step 4. Thiolysis. Finally, the enzyme Thiolysis cleaves the molecule with coenzyme A, releasing 1 acetyl-CoA and a fatty acyl-CoA shortened by 2 carbons. This shortened fatty acyl-CoA re-enters the cycle, and the process repeats until the fatty acid is completely broken down. Thanks for listening. Like, share and follow the channel to keep learning biochemistry the clinical way.